Hello everyone, my name is Jung Hun Oh, working at Memorial Sloan Kettering Cancer Center. I'm glad to present our work in ISMB. The title of our work is PathCNN, Interpretable Convolutional Neural Networks for Survival Prediction and Pathway Analysis applied to glioblastoma. CNN has achieved great success in the field of image processing, dealing with grid-structured inputs or images and efficiently capturing local dependencies. However, in the field of bioinformatics, rather than CNNs, traditional neural networks have been extensively employed due to non-grid structured format typically represented in biological array data. Even the lack of interpretability is a crucial factor that limits the adoption of neural networks in the field of medicine. To increase the interpretability of classical CNNs applied to biological data, we propose to use pathway image in connection with gradient-weighted class activation mapping, Grad-CAM. I'm going to demonstrate the method applied to predicting long-term survival in patients diagnosed and treated for glioblastoma, GBM. GBM is one of the most malignant tumors with poor prognosis and outcomes, with median survival of 14 months after diagnosis. For this study, we downloaded multiomics data from cBioportal database, including gene expression, profiles, copy number variation, and methylation. Around 8,000 genes were common in the three omics types. We defined long-term survival, LTS, survival larger than 2 years. In our data, 55 patients belong to LTS group. Non-LTS was defined as survival less than 2 years. 232 patients belong to this group. We employed CAG database, removing disease-specific pathways, resulting in 146 pathways. To generate pathway images, we employed the following approach. This is an example. So there is a pathway and associated genes. We extracted the gene expression profiles for those genes here. And then we conducted the principal component analysis, PCA, to reduce the dimensionality. We repeated this process for individual pathways. More specifically, this bar indicates a set of principal component pieces for a given pathway. We do the PCA analysis for individual omics types. After the process, we rearrange the data for a given sample, and we combine the pieces for multiomics data. The row is pathway, and the column is corresponding pieces. At the end, a sample is represented by a matrix. We call it pathway image. Here, each pixel has meaning, which is biological pathway. Our goal is twofold. One is to build a model of long-term survival in GBM. And the second goal is to identify important pathways relevant to survival. So this is interpretable CNN. We try to place the relevant pathways nearby to maximize the capability of CNN modeling, which use localized information on images. Doing that, the Grad-CAM can also provide better localization mapping. So two most correlated pathways were placed on the top two rows on the pathway images. Then a pathway that was most correlated with the pathway in the second row was placed on the third row. So this process was repeated for all 146 pathways. Our CNN architecture consists of two convolutional layers, followed by max-pooling and dropout layer, and then fully connected layer, one more dropout layer, and up layer. We added age variable into fully connected layer. Age is important clinical variable in GBM. I will describe this later. After modeling, for biological interpretation, we employed Grad-CAM. Grad-CAM uses gradient information flowing into the last convolutional layer of the CNN to obtain the class discriminative localization map. We first compute the gradient of score for class C, YC, with respect to feature map AK. K is index. An average value of this feature map was used for weighting value for the feature map. Then we computed the weighted combination of feature maps followed by ReLU, which is Grad-CAM. In the classification of cat and dog, as you can see for dog, the face was highlighted, and for the cat classification, the body was more highlighted. Grad-CAM is generalizable version of CAM, but the concept is very similar. I brought this CAM example for clear explanation about how to generate activation map. As you can see, after getting weighting values, the class activation map is generated by computing the weighted sum of feature maps. This figure illustrates how to use Grad-CAM in our study for biological analysis. In the last convolutional layer, we compute gradient of LTS with respect to feature map AK. Average value of all pixels is used as weighting value of the feature map AK. Then we compute weighted sum of the feature maps, which is Grad-CAM. We do this for each class of each sample. So each sample generates two Grad-CAM. Unlike other images, in our pathway image, each pixel has fixed unique biological meaning. So for statistical analysis, we compute the difference between two Grad-CAM for a given sample. So this figure B shows the matrix for each sample in LTS and the non-LTS groups. Using Wilcoxon-Necker sum test, p-value of each pixel was computed between LTS and the non-LTS groups. So this information was used for biological interpretation of pathways. There was a significant difference in age between LTS and non-LTS groups. IDH wild-type GBM is more common and has worse prognosis, whereas IDH mutant GBM is more frequently observed in younger patients and has been associated with longer survival. In our data, there were IDH mutations in only 6 and 2 cases in the LTS and non-LTS groups respectively. However, the difference was statistically significant with p-value 0.0013. For modeling, we used 5-fold cross-validation. After the repeating this 5-fold cross-validation 30 times, the average area under the curve, AUC, was reported. In each 5-fold cross-validation, 20% of samples were used for testing. The remaining data were further split into 80% of samples for training and 20% of samples for validation. This graph shows the result of our modeling with different number of pieces in pathway images. When 2 pieces were used in the pathway image, the performance was saturated with an average AUC of 0.753. A combination of two omics types was tested, a combination of gene expression and CMB, and a combination of CMB and DNA methylation achieved comparable performance. But a combination of gene expression and methylation showed much worse performance. In addition, the CNN modeling of pathway images with a single omics type, best performance was achieved in CMB, followed by gene expression. In addition to GBM, we compared the performance of our method with other alternative methods on multiple cancers, including low-grade glioma, lung cancer, and kidney cancer. Our approach achieved best performance in GBM, lung cancer, and kidney cancer. However, in LGG, SBM obtained the best performance. This is pathway image with the statistical test result. Red color indicates pixel or pathway with adjusted p-value less than 0.001. As you can see, significant pathways are clustered. This is representation in table format. For the pathways in the box, the Kaplan-Meier analysis was performed. The samples were dichotomized by median split of PC1 in gene expression for the 4 pathways. All had statistical significance. Through the literature review, these pathways have been found to be relevant to GBM. Conclusion. In this study, we proposed the PathCNN, a novel algorithm to build interpretable CNN models based on the concept of a pathway image generated using multi-omics data. The model was able to predict long-term survival of GBM patients better than other alternative machine learning methods. The application of Grad-CAM directly on the pathway image enabled the identification of plausible pathways that affect the survival in GBM. We appreciate funding agencies. Thank you for your attention.